This is podcast number 51. And all week, this week, we're talking about emergent properties. And this chapter deals with how cells get together for emergent properties. This particular example of emergent property is the ability to tell time. So it's been known since the 1970s that the daily cycles, circadian cycles that are seen in cells are based in genetics. That was a pretty wild idea back in the 70s. Now it's well established, and that is an emergent property of cells. So some examples you might be familiar with, there are flowers such as morning glories that bloom in the morning and then they are shut down by mid-afternoon, like this. In Drosophila, which is the fruit fly, this is a model organism for studying circadian rhythms in animals. Sometimes people have trouble pronouncing this word. It's Drosophila, not Drosophila, but Drosophila. And these are fruit flies like the that are attracted to rotten fruit. They're little tiny guys, and they are easy to raise in the lab. And this is how they hatch. The adult flies hatch, and they hatch at dawn into an adult. And the group that established the finding the genes that are responsible for the circadian rhythms first started off with a big group of flies that they mutated on purpose. So they fed them a chemical that mutated their DNA, and they knew that they were going to do that. And they thought that, okay, we're going to look at this behavior, which we can predict, and this is when the flies hatch. So this is time of day, over the course of a day, 24 hours. So time 0 is at the beginning of one period of the day all the way to the end of time 1. This is 24 hours later. So that early in the morning you see flies hatch, and then no hatchings toward the end of the day, and then the beginning of the next day, and so on. And this is in an unmutated fly. And then they made a whole bunch of mutations in these flies, and they thought if it's genetically based, we should be able to find some flies that don't follow this pattern. And they screened a lot of flies to find them, 2,000 of them. And they were able to identify some mutations, and they were called PER mutations, PER, short for period, and that they have a different period. So if you look at the size of this bar and that what is the inactive period for hatching in wild-type flies, it's shorter in the PER-S mutation. It is longer in the PER-L mutation, S for short, L for long, and for the PER-O mutation or PER-0 mutation, it's like they don't have any rhythm at all, that they just hatch whenever they want. So again, here's the wild type cycle, and this is the period of inactivity when they're not hatching. The period when they're not hatching is shorter in PER-S, longer in PER-L, and then no period at all in PER-0. So this is a pretty big deal, identifying a gene that controls this basic aspect of behavior. That was a pretty big deal back in the 70s. Part of what they wanted to do was to find out, does it affect all kinds of periodic behavior? So there's hatching, but then there's also just when are the flies active? So there was a postdoc in the lab who set up a experimental system where the flies crossed an invisible beam of light, invisible to the flies. This is similar to you walking into a Home Depot that breaks a infrared beam of light and the doors open. So every time the flies walked across this beam of light, it's recorded as a little black mark on here. And then if the flies aren't moving, you get a flat line. So you can see in wild type that, and each line here is a different fly. You get ideas of that they're active for part of the day and not active for other parts of the day. In per zero flies, they're active or not active all parts of the day. In per S, their period of activity is, the cycle of the period of activity is shorter, and per L, it's longer. So what are these mutations? They are called SNPs. SNP stands for single nucleotide polymorphism. So single nucleotide, just one nucleotide is changed between the wild type and the mutant. And then polymorphism means that in a population, you can have different individuals with different nucleotides at that particular site. By the way, this is what they measure if you send away your DNA to 23andMe or Ancestry.com. They're not actually sequencing your whole DNA. They're just measuring a whole bunch of SNPs that are known in humans, and that we know humans vary at this spot and in this spot and this spot. And so we're just going to look at those spots and see what kind of variation you have when you measure that. Okay, so these are different changes. In the case of per zero, it turns it into a stop codon so that the protein is not made. In the case of per S, it changes the amino acid so the protein has a different shape. And per L, it changes it into a different amino acid so protein has a different shape. And that has consequences in terms of how the protein works. So this research went along, and there's still lots of research going on in fruit flies about periodicity, circadian rhythms. And for a long time, 10 years or so, it was thought that only multicellular eukaryotes had daily cycles, and you wouldn't see that in prokaryotes. And then another lab found daily cycles in cyanobacteria, which is a prokaryote. So they measured when the cells divide over the course of days, and the number of cell divisions in a population is lowest right at dusk. And then you can see that over and over again over a period of days. So there's definite periodicity here. And you can actually do statistics on this to see, are you actually seeing significant periodicity or not? We did this in the Tahiti class. We looked at fish that change color, and their color change with pulse on and off like this. And then we use those statistics to find out, is this significant periodicity or just random changes? And it was significant, and we got that published. Undergrads doing that work in Tahiti. Okay, so in the cyanobacteria, the work has progressed well and figured out the molecular mechanism for how this works. And the protein that is the timekeeper is called Chi, and there are different subunits of the protein. It's a protein with quaternary structure, and it has A, B, and C subunits. So this is a top-down view. This is a slice through the side, and these ATP molecules are part of the Chi protein. You can't see them very well if you're looking for the top-down front, but if you slice through the middle, you can see them well. And the A, B, and C proteins are important to how this cycle works. We'll walk through that. So you can think of Chi A, the A subunit, as adding phosphates, okay, from ATP. and Chi B as removing phosphates, or B for biphosphates. I know that's pretty hokey, but hopefully it'll help you remember. And they compete with each other throughout the period of a day. So you start off with Chi C, that's at dawn, no phosphates on it. And then Chi A binds to the protein, and Chi B leaves. Chi A activates Chi C, which is a kinase. Kinases, we know from previous podcasts that kinases take the phosphate from ATP and add it onto proteins. In this case, it's added onto this Chi protein. And as the cyanobacteria go through their day, they get more and more phosphorylated and more proteins are being added. Chi B rejoins and starts to compete with Chi A for binding to the protein, the Chi protein. And Chi B is removing phosphates. So as Chi B starts to win in the competition, it starts to lose those phosphates. Chi B activates Chi C phosphatase. Okay, so there's Chi C kinase and Chi C phosphatase. Chi C phosphatase is removing the phosphates, and then you're back at dawn where you have no phosphates. So throughout the course of the day, you accumulate phosphates. That changes how the proteins that are making the decision on whether or not the cell should divide, how they behave. And then you get back to dawn, there's no phosphate, and the cell makes a decision. And so this pattern of getting phosphorylated and then dephosphorylated throughout the day is what's responsible for the periodicity, or this 24-hour cycle. So, but why are there circadian rhythms? Are they selectively advantage? How do they give? Do they give a selective advantage, or is it just something that organisms do? So one thing about single cell organisms, or at least really small organisms, is that have a short generation time, it's relatively simple to do evolution experiments and have them compete with each other like in natural selection experiments. So with these cyanobacteria, they started with a population that was 90% with a mutation where they did not have the periodicity. So they weren't using Chi to set up that 24-hour rhythm of reproduction. And then 10% of the population was wild type, and said, under conditions of constant light, who does better? And the mutant actually does better. So they outcompete cells in the wild type, and the wild type lose. They go from 10% to like no percent, close to no percent. And the per zero, the guys in purple here, lose. But if you redo the experiment again, and now you grow them in a light-dark cycle, like 12 hours light, 12 hours dark, approximating what these organisms would see in the ocean, and these are ocean-living bacteria. What you see is the per zeros cells decrease to about 30% of the population from 90% of the population, and the wild type win in that situation. So under conditions like we have on Earth, where we have light and dark cycles, it is maladaptive to not have a cycling sort of system that pays attention to that light-dark cycle, at least in this species. Okay. They also wanted to see what other abiotic, meaning not caused by biology, factors in the environment may influence the clock. And another one of those is obviously temperature. So if you think about things that pay attention to seasons, they pay attention to how much light is out there, but also the temperature. So if you think about leaves changing color, or leaves staying on trees or dropping off trees, or just when things grow, they pay attention to light and also color So they are light and also temperature, sorry. So they wanted to see, can temperature influence how these clocks run? And so they did an experiment where they have the bacteria going through their cycle, and they're normally grown at a pretty warm temperature, 30 degrees C. But then they add in a pulse of cold water for about 6 hours and say, how does that affect the cycle? Okay. So at 30 degrees C, where they don't add the cold water, you get their standard up-down cycle over approximately 24 hours. Okay. They add in the shock in that little purple bar there for 6 hours, centered at starting 3 hours before, 12 hours into the cycle, and 3 hours after, 12 hours into the cycle. And you see the circadian rhythm slightly shifted to the right. So you're slowing down these cells, and the rhythm is slightly shifted to the right. Okay. And if you continue that over days, the shift becomes more pronounced over time. And if you keep on going, it gets more and more shifted. Okay. So how these authors interpret these data is that if you have the wild type situation at 30 degrees and light dark cycle 12 hours, the term for these cycles is called circadian, and it means circa-adian, which means around 24-hour cycle. And it's not exactly 24 hours. So if you look at the trough in each one of these peaks, this is right about 24 hours. This is not quite 24 hours after that. And this is not quite 24 hours after that. So it's sort of like you got a watch that is losing a little bit of time every day. If you have a cycle that can get reset, so to speak, with cold, with temperature, then you can reset your clock every day. So if, for example, if you know that the ocean will be colder after coldest right before dawn, so you've had a whole night of no sun, it's getting colder and colder and colder as the night continues, and right before the sun comes up, that's when you get the coldest temperature. If you look at your weather app and you look at the hour by hour temperature, it's usually really the coldest right before the sun comes up. So, and that's not going to be the same year round. So if it's important or adaptive for you as an organism to know when should I be active? Well, the sun is not coming up at the same time year round. We know that living in Ohio. Sunrise is not the same time year round. And when the best part of the day in terms of temperature and sunlight is, it's not going to be the same year round. So if you can pay attention to both temperature and sunlight and sort of reset your clock every day, you have a better chance at being active in the most beneficial part of the day. So like when your prey are active, or when it's best to photosynthesize, that sort of thing. So reacting to temperature is a way to reset the clock with seasons. Now, obviously, there's lots more to figure out about how this periodicity works, but it's really interesting research and really important and applicable, even though these are cyanobacteria that live in the ocean. If we understand more about how this periodicity works, we can understand more about why is it that daylight savings time messes humans up so much. because your biology is still on a circadian clock and you have shifted your electronic clock, but not your biological clock, and it takes a long time for you to shift after that. Or why is it that people who work second shift or third shift, work through the night, can have health problems? It's because you're bucking your evolutionarily determined active period. And you're saying, I'm going to be active when most humans are not active, and that can have health consequences.